Today, I will be talking about fine mapping causal tissues and genes at disease-associated loci. And a bit of an outline for our talk today. I'm going to start with some motivation. So why do we want to fine map causal tissues and genes at disease loci? Then we're going to talk about the methods we developed to do this task. We're going to evaluate those methods in simulations. And then we're going to show results applying this new method of tissue gene fine mapping to 45 UK Biobank diseases and complex traits using 38 GTEx tissues. But to start, we're going to start with motivation. And to motivate this, there's been a tremendous amount of work recently showing that genetic predisposition for complex diseases act through disease-relevant tissues and cell types. And here we're showing a plot from Finucane et al., Nature Genetics 2018, showing specific cell types and tissues are enriched for certain diseases and complex traits. And oftentimes, the enriched tissues are disease-relevant or critical tissues. As you can see, for LDL, you get liver popping up strongly. However, all this analysis that's been done recently has focused on integrating information across the entire genome. And what we're going to try and do in this talk is focus in on a given region of the genome and zoom in on a given locus and try to identify what the causal tissue is underlying that specific disease locus and whether or not there's a causal gene that mediates that disease signal in that tissue. So a bit more detailed, the goal of this project is to fine map causal tissues and genes at disease signals. And here we show a cartoon Manhattan plot showing a given GWAS locus. And what you can see here in the cartoon is there's hypothetically 3 different causal signals underlying the disease locus. And as you may imagine, what could happen here is this first signal is mediated through a given gene. We're going to call it gene 1 in the tissue, adipose tissue. And then there's potentially gene 2 mediating the disease signal in brain. And then there's this third signal that doesn't go through any gene in any of our assay tissues. And we're going to call this a non-mediated variant, meaning their disease signal doesn't go through any any assay tissue or gene. And a bit more concretely, we're going to develop a statistical model that we're going to call TGFM, or standing for tissue gene fine mapping. And it's going to fine map the causal tissue and gene and each genetic disease signal. And what this is going to involve is going to involve calculating causal probabilities for each gene tissue pair and the probability for each non-mediated variant. And we're going to do this based on the integration of GWAS data and EQTL data from diverse tissues. Okay, jumping into the methods. I'm going to start with what I think is potentially a naive way and not what we actually end up doing to identify what the causal tissue and gene is, but it's going to ultimately motivate the methods underlying TGFM, or tissue gene fine mapping. And what you can do is you could run a transcriptome-wide association study, or a TWAS, to see whether a given gene in a given tissue, in this case, we're looking at gene 1 in blood, and whether or not gene 1 in blood is associated disease. And the way a TWAS assesses whether gene 1 in blood is associated disease is through this two-step process. In the first step, what you do is identify cis variants with non-zero effects on the given gene using EQTL data. And then given those predicted genetic gene expression measurements of gene 1 in blood, you can evaluate whether that is associated with disease or not. However, this has a bunch of caveats. And most prominently, TWAS associations cannot distinguish causal from tagging gene tissue pairs. And this is analogous to variant linkage disequilibrium obscuring causal from tagging variants if you just use marginal GWAS association statistics. And more specifically, a significant TWAS association for a given gene tissue pair can result from 2 things. The first, which you may desire, is the gene tissue pair having a causal effect on the disease, which is hopefully what you want. But it can also originate from the gene tissue pair not having a causal effect on disease, but it's just simply correlated with another gene tissue pair that is causal or a non-expression mediated genetic variant. And so what TGFM is going to do is it's going to try and distinguish these causal from tagging gene tissue pairs. And the way it's going to do this is a similar 2-step process, where in the first step, you identify cis variants with non-zero effects on each gene tissue pair using multi-tissue EQTL data. So you build this mapping from variants to genes. Then in the second step, you use a multi-variable regression approach to identify which genes have a causal effect on disease when considering all the genes. And in addition to that, considering all effects of a given genetic variant that doesn't go through expression and just goes through its own pathway. And so more specifically, and in terms of the underlying mathematical model underlying TGFM, we have this trait generative model where our trait Y is equal to the sum of effects from two different genetic effects. We first have these non-mediated genetic effects where G is our genotype matrix and beta is the vector of causal non-mediated variant to trait effect sizes. And then we have this gene-mediated effect, genetic effects terms, where we have an effect from each gene tissue pair where G indexes genes and T indexes tissues. Again, G is our genotype matrix. Delta GT is the causal eQTL effect sizes. So G times delta GT is the predicted gene expression in gene G and tissue T. And then alpha GT is the causal effect of gene G and tissue T on the trait. And ultimately, what we'd like to do is we'd like to learn about this beta vector and this alpha vector. The beta is the vector of causal non-mediated variant to trait effects, and alpha GT across genes and tissues are these gene to trait effects in a specific gene and specific tissue. And what we're going to do to try to identify which elements of this joint causal effect vector of beta and alpha are non-zero is we're going to take a fine mapping approach, and we're going to use the previously built model of SUSY fine mapping, which assumes that basically these causal effects, beta and alpha, is equal to the sum of these sparse effects of single variants. And we're going to assume there are L single genetic elements, where a genetic element is either a variant or a gene tissue pair. And we're going to assume they follow this joint probability distribution. And specifically, we're going to assume TGFM fine maps L disease components in a given genomic region. And in addition to that, TGFM is going to estimate a posterior distribution on this latent variable gamma L. And what gamma L represents is the categorical probability that the trait signal L comes from one of the K variants or one of the G times T gene tissue pairs. And once we calculate a posterior distribution on all the gamma Ls, we can compute this quantity that we're going to talk a lot about today, which is a posterior inclusion probability, or a PIP. And a PIP basically just captures the probability that each genetic element has non-zero effect on the trait. In addition to that, we integrate some additional features into TGFM. First, we learn a genome-wide parameter pi, which represents the probability across the genome that each genetic element is causal. And it does it by integrating non-mediated variants together into one class and genes from a specific tissue together into one class. So it's this tissue-specific prior. In addition to this, previous methods that do gene fine mapping do not account for uncertainty in our predicted causal eQTL effect sizes, which is delta GT. And while I don't have time to get into the details, TGFM uses a sampling approach to model uncertainty in the causal eQTL effect sizes, delta GT. Okay, so that was the technical details of our methods. And if that went over your head, this is kind of the short summary of what TGFM does. It takes this input, GWAS summary statistics, in simple variant LD, so correlations between all the SNPs, and multi-tissue cell type and eQTL data. And what it's going to output is a PIP, or a posterior inclusion probability, for each gene tissue pair, and a PIP for each non-mediated variant. In addition to this, once we have the PIPs for each gene tissue pair, we can aggregate or collapse them into gene level PIPs as well. So we have these three different versions of PIPs, one for gene tissue pairs, one for genes, and one for non-mediated variants. And we're going to refer to these three different things, variants, genes, and gene tissue pairs, as quote unquote genetic elements for the rest of this talk. Okay, next I'm going to jump into simulations. And first what we do is we compare TGFM to two existing approaches that do some form of gene fine mapping. One is called FOCUS and one is called COLOQ. And while FOCUS and COLOQ, neither of them were built to do tissue gene fine mapping, they were built to try to identify whether or not the given gene is causal. In the case of FOCUS, it could be immediately extended to the case of tissue gene fine mapping, which we call FOCUS-TG. So we compare TGFM, which is in red, to these three alternative approaches, and we look at how calibrated they are in identifying causal gene tissue pairs in simulations. And what you can see at two different PIP thresholds, PIP of 0.5 on the left and PIP of 0.9 on the right, TGFM is well calibrated, and in cases across varying eQTL sample sizes on the x-axis produces well-calibrated posterior inclusion probabilities, where existing approaches, FOCUS-TG and FOCUS, as well as COLOQ, result in miscalibrated PIPs. And I don't have quite time to get into the details as to So why this happens, but there's two main reasons. First is that Focus and Colloq do not model the effect of non-mediated genetic variants and just consider gene tissue pairs. And by modeling non-mediated variants, we're able to distinguish causal gene tissue pairs from causal non-mediated variants, which these existing approaches can't do. And in addition, there's an additional step that, as I said in the methods, TGFM models uncertainty in predicted genetic gene expression, which is a new feature that these existing approaches do not do, and that improves our calibration as well. Next, what we do is we just focus on TGFM, and we look at how calibrated the PIPs are in terms of gene tissue pair fine mapping shown in red, gene fine mapping shown in green, and non-mediated variant fine mapping shown in blue. And what you can see is TGFM for all these genetic elements is well calibrated at various eQTL sample sizes on the x-axis and various PIP thresholds X and Y on the left and right. In addition to that, here we're showing not calibration, but how TGFM does at fine mapping, gene tissue pairs in red, genes in green, and non-mediated variants in blue at two different PIP thresholds and what power it has, which we're showing on the y-axis. And what you can see is power to fine map gene tissue pairs shown in red and genes shown in green increases with eQTL sample size, which is expected. As you have more eQTL samples, you should be more confident to fine map which gene tissue pair is causal. But as you can see, the ability to fine map a non-mediated variant is independent of eQTL sample size. Okay, next we're going to jump into real results. So we're going to apply TGFM to fine map 45 UK Biobank diseases and complex traits using 38 GTEx tissues. First, we just summarize the results. So how many fine map genetic elements do we get across all 45 traits? Here we're showing for a subset of traits, 16 genetically independent traits. And as you can see, on average, TGFM identifies 16 gene tissue pairs per trait at PIP greater than 0.5. TGFM identifies 62 genes per trait at PIP greater than 0.5 and 131 variants per trait at PIP greater than 0.5. And here we just show a neat example of a specific locus that TGFM fine maps. And this is for the trait eczema on a specific locus on chromosome 11. And what we're showing is the marginal GWAS and TWAS negative log 10 p-values on the y-axis of each genetic element, where the blue circles are these variants and the red triangles are gene tissue pairs, where gene tissue pairs are centered based on their gene's TSS. And what you can see, and these dashed lines here, the red is the TWAS genome-wide significant threshold, and the blue is the GWAS genome-wide significant threshold. And what you can see is there are a lot of variants and a lot of gene tissue pairs that are genome-wide significant. However, if you just look at the PIPs, which is based on these coloring of the points, TGFM is able to localize all the signal from all these significant marginal associations to a single gene in a single tissue. In this case, it's OVAL1 in lymphocytes, and it's PIP 0.75. And OVAL1 is kind of this well-studied gene for autoimmune diseases, and it's thought to work through immune-mediated responses. The lymphocyte is the relevant tissue here. And here we just have another example, and this is for the trait hypertension, and we get the well-known hypertension gene ACE, and in this case, it's identified in adrenal gland. Okay, and next we can kind of zoom out, and we can look at, for a given trait, of all the fine map gene tissue pairs, which tissues do they come from? And oftentimes what we found is TGFM implicated gene tissue disease triplets in disease critical tissues. And as we can see, basically all of the gene tissue pairs identified for LDL direct come from either liver or whole blood. For all autoimmune diseases, almost all of them come from spleen and lymphocytes. Vitamin D levels coming from skin, systolic blood pressure coming from artery aorta, artery tibial, and brain cerebellum. Next, what we did is we looked at whether the genes we identified made sense, and what we do is we intersect TGFM gene PIPs with this previously generated gene metric of POP scores, and what we can see is there's near monotonic increase of the POP scores with TGFM PIP. And lastly, what we do is we apply TGFM to not fine map gene tissue pairs, but to fine map PBMC cell types. So in this case, we use single cell eQTL data from 9 immune cell types. We do the same thing, but instead of just looking for gene tissue pairs, we look at gene cell type pairs based on these fine-grained cell types. And what you can see is we're able to help resolve biological loci using the single cell EQTL data that could not be resolved using bulk tissue. And so this is for the trait monocyte count. And we identify here a gene called CD52 in the cell type classical monocytes. And in general, what we found is we identified 30 additional fine map gene cell type pairs using this approach across all traits. Most of them came from blood or immune traits as opposed to other traits. And in addition to that, oftentimes the TGFM implicated gene cell type triplets, cell type disease triplets, are concentrated in disease critical cell types. So for example, for monocyte count, we're getting pretty much all of them coming from non-classical monocytes and classical monocytes. For lymphocytes, mostly from T4s, and from all autoimmune, mostly coming from T8. So in conclusion, we developed a new model, TGFM, to jointly fine map the causal gene and tissue at disease loci. TGFM generates well-calibrated tips for gene tissue pairs, genes, and non-mediated variants in simulations. High-pip gene tissue pairs often originate in disease-relevant tissues, and high-pip genes are enriched in independent gene sets. And TGFM applied to fine-grained cell types and single-cell EQTL data can help resolve the biological mechanism underlying disease loci. In conclusion, I'd like to thank Alkes Price and a few other members of the Price Lab, including Martin Zhang, Tiffany Amaruta, and Jordan Rosen. Thank you very much.
I'm going to be talking about approaches to account for multiple births in epidemiological studies of pregnancy. And multiple births sort of account for approximately 3% of live births. And these are sort of twin pregnancies, but a smaller percentage are higher order multiple births. And multiple gestation itself can be sort of an exposure, a mediator, an outcome, a confounder. So it can play different roles depending upon the particular study question. And in this presentation, I'm going to focus on the setting when multiple gestation is an intermediate variable between the exposure and the outcome. And the aim is to sort of compare different strategies for accounting for multiples in perinatal epidemiology through both Monte Carlo simulations and also an applied example evaluating the risk of malformations with clomiphene use. Clomiphene being a medication used to treat infertility and being sort of associated strongly with multiple gestation. In terms of the sort of role of multiple gestation and multiple births in sort of perinatal epidemiological studies, when it's an intermediate variable, it could have sort of both a statistical role in terms of sort of introducing clustering. There have been sort of studies looking at they've looked at the intra-class correlation coefficient and found sort of coefficients between 0.13 and 0.79 in different RCTs with different outcomes in pregnancy. But also from a sort of structural perspective, sort of causal perspective, multiple gestation may sort of interact with the treatment, and it could also be a sort of mediator of the sort of treatment outcome association. And here you can see a sort of couple of causal directed acyclic graphs with a sort of multiple gestation as potentially a mediator, and also with potential confounders L1 and L2 of the treatment, multiple gestation, and multiple gestation outcome associations. And the approaches that I'm sort of comparing are some common approaches that sort of have been applied in perinatal epidemiology. So that includes sort of restricting to singletons, counting sort of outcomes at the pregnancy level. So at the pregnancy level, does the mother have one or more infants with, for example, a malformation, if malformations are the outcome? And then different approaches that including all the infants. So without sort of consideration of clustering, using sort of generalized estimated equations to account for clustering with different working correlation matrices, with sort of independence or exchangeable working correlation matrix. And these actually, the difference with these different working correlation matrices lead to slightly different estimands, with the sort of independence working correlation matrix being the same sort of estimand as just a sort of approach without consideration of clustering. And I guess in some way sort of restricting to singletons or counting outcomes at the pregnancy level does eliminate any sort of issue to do with clustering, but could sort of lead to sort of different estimands. And in some cases, in sort of data that we have in practice, it may be hard to determine whether outcomes are in one infant or in the other infant, and therefore we may be sort of limited to applying this one of these two sort of approaches of restriction or sort of counting outcomes at the pregnancy level, when sort of the question of interest may be actually at the infant level. And for the simulations, we had different data generating mechanisms, and we used parametric models to simulate the variables being treatment, multiple gestation, and outcome. And for the simulation, we considered multiple gestation as only sort of a twin pregnancy, so we didn't consider pregnancies of higher order. And we induced correlation within the sort of clusters as well. And we used sort of log linear models to simulate the multiple gestation and the outcome. And in terms of the parameters, we sort of fixed a number of parameters, such as prevalence of the treatment at 50%, prevalence of twin pregnancy at 2%, amongst untreated pregnancies, the prevalence of outcome at 3%, and an intra-class correlation coefficient of 0.3. And these are sort of some values that we might sort of see in practice, for example, of the prevalence of twin pregnancies, and of the sort of prevalence of an outcome such as congenital malformations. And in the simulations, we vary the strength of the treatment multiple gestation association, as well as the associations between multiple gestation and outcome, and the sort of direct treatment outcome association. The analysis was sort of Poisson models with robust standard errors to estimate risk ratios, and also sort of generalized estimating equations with sort of cluster robust standard errors. And here's the sort of first result from the simulations. So we can see that I guess the direct effect is a risk ratio of 2, but here with the sort of naive analysis being a sort of an infant level analysis without adjustment for clustering, we see sort of that as the sort of treatment multiples association increases, because there's a multiples outcome association of 2, we start to see sort of an increased association due parts are mediated by this multiple gestation. And now if we add the sort of GE independence approach, we see they're sort of estimating the same estimand, and we're getting very similar estimates. And if we add the sort of exchangeable generalized estimating equations approach, we see that it sort of estimates slightly different estimand because it's sort of weighting those weighting the multiples differently. And if we look at the pregnancy level, we see also that slightly different, but again, these estimates aren't greatly different from each other. And if we think about it, when we're sort of counting if one of two sort of a twin sort of has an outcome, then the risk at the sort of pregnancy level will almost be sort of double the risk at the infant level amongst those twins if their sort of outcomes are independent, as we can see in this sort of calculation. But we also sort of effectively downweight those multiples in the sort of pregnancy level, because we only count sort of one of the two when doing that sort of pregnancy level analysis. So it leads to fairly similar estimates, at least in this scenario. And we're sort of increasing, or perhaps even introducing, association between multiple gestation and outcome when counting at this pregnancy level. But we're also sort of potentially reducing the association between treatment and multiple gestation. And here we can see sort of like the direct effect when restricting to singletons of 2. But depending upon sort of in practice, we might not actually be sort of estimating a sort of controlled direct effect in mediation terminology, but there may be sort of confounding there. And sort of when we're restricting to singletons, we may even introduce an association where there isn't one overall due to sort of a collider, well, multiple gestation being a collider. And now we compare this when there is no multiple gestation outcome association. So in effect, there is no sort of mediated association. And we could see that the approaches all are fairly similar, with the exception of the sort of pregnancy level analysis. And then if we have the treatment outcome association as null as well, it's fairly similar to our first graph. We are getting this sort of mediated association, and we see slight differences between the approaches, with the largest difference being between the restriction to singletons versus the other approaches. And then we also have the sort of when both the treatment outcome association among singletons or sort of also amongst multiples is 1, and the sort of multiples outcome association is also 1, and we see this sort of very similar estimates with some slight difference again with the pregnancy level. And now we have the sort of empirical standard errors. So how much variance are the estimates having across the simulations? And we see that they have very similar variance, different estimates, with the exception of the restriction to sort of singletons, which we might expect because we've sort of reduced the sample size in that approach. And we can also compare the sort of model standard errors to the empirical standard error here and see sort of, is the model standard error too small? And we can see that the approaches have fairly similar sort of relative error in the model standard error, with the sort of, if you will, naive approach doing analysis without taking into account clustering at the infant level, having slightly less sort of slightly larger standard errors relative to what they slightly smaller standard errors relative to what they should be. Yeah. And now we go to an applied example. So clomiphene is a medication used to treat infertility. It's strongly associated with an increase in multiple gestations, and there is some evidence for a weak association with some malformations, but perhaps some of that association may be also mediated by multiple gestations. And the design was a cohort study of pregnancies ending in live birth, identified in MarketScan insurance claims database between 2010 and 2015. We selected mothers aged 12 to 55 years and those pregnancies ending in live birth amongst those mothers. And the exposure was clomiphene use versus non-use, with an outcome of major congenital malformations. And we adjusted for sort of potential confounders of the sort of treatment outcome and multiple gestation outcome associations. And similarly to before, we used Poisson models to estimate risk ratios. And we had the sort of approaches that you don't need to, that sort of eliminate the issue of clustering with the restriction to singletons or counting outcomes at the maternal level, but slightly different estimands. And also the sort of approaches including all infants, so without considering clustering, or using sort of GEE approaches, either independence or exchangeable working correlation matrices. And additionally, in this applied example, we also have a mediation analysis to estimate a pure direct effect and total indirect effect. And looking at the baseline characteristics, we have about 400,000 live births. And we can actually see that from this table that clomiphene users have a sort of higher percentage of multiple gestation, so 13% versus 4.4% for non-use. And we can also see that sort of diabetes, which is potentially a sort of major confounder, is more prevalent amongst clomiphene users than non-users. Diabetes being sort of associated with a number of congenital malformations including cardiac malformations. And sort of in our results, we find that sort of treatment is strongly associated with multiples. So we find a sort of adjusted risk ratio of 2.45, and that multiples are associated with congenital malformations with an adjusted risk ratio of 1.54. So potentially, multiple gestation is a potential sort of mediator for treatment outcome association. And here we have sort of a figure with some of the results. And what we'll actually see is that the different approaches, such as pregnancy level and the infant level approaches, all in this particular example sort of result in similar estimates. So you have sort of adjusted risk ratios of about 1.16 or 1.15. But when we're sort of restricting to singletons, we have a slightly sort of smaller effect estimate of 1.07. But again, all these sort of risk ratios are quite small, so there's a possibility that they could be due to residual sort of confounding, and sort of having that sort of adjusted risk ratio of 1.07 in singletons suggests there may not be a direct effect, at least amongst those without multiple gestation. Of course, the sort of, if we assume that all the assumptions of a controlled direct effect are met, then it may be that there's sort of interaction, or there may be sort of mediation there. And if we add the sort of mediation analysis, we can see that sort of for the pure direct effects, the estimated risk ratio is 1.09 with a sort of confidence interval touching the null. And we also have some evidence for an indirect effect, but again, the sort of association is quite small at 1.05. So in general, there's not sort of strong evidence for like a strong association between clomiphene use and malformations. And some of the association that is there may be due to multiple gestation. And some challenges and limitations. Well, it's sort of difficult to assign outcomes to specific infants in claims data. And in some cases, depending upon the outcome, we may need to be limited to approach restricting to singletons or to counting at the pregnancy level, which sort of we need to take into account because it may be slightly different estimand. There is in these sort of studies, when we're restricting to live births, the possibility of selection bias. For the sort of mediation analysis approaches, there are sort of quite strong assumptions for estimating pure direct and total indirect effects, such as sort of a very strong cross-world independence assumption that we may not feel is met, and sort of no confounding of the mediator treatment and mediator outcome associations. And we also think there could be some residual confounding in this case. I mean, in this case, we use as an example to show the sort of issues around multiples. If we're sort of, when we're sort of doing a study more for sort of looking at actual safety, then we might want to compare to another drug used for infertility. And in a study conducted by my colleagues using a target trial, they compared clomiphene relative to letrozole for the risk of malformations, and find sort of lower risk of malformations with clomiphene relative to letrozole, which is sort of possibly mediated by multiple gestation, with multiple gestation being increased amongst those dispensed letrozole versus clomiphene. And some conclusions. It's important to consider the estimand of interest when deciding an analytic approach. And it sort of depends upon the potential question that we may be asking. So what is the sort of overall effect of treatment on the infant level risk of malformation? Then maybe we want to use a sort of infant level analysis, I guess ideally accounting for clustering, although in some cases that accounting for clustering may not have a large effect, given that sort of multiples are quite a small percentage of the cohort in a lot of cases. And then also sort of what is the overall effect on the risk of the mother having one or more infants with malformation. So that may be an interesting question to inform a sort of potential mother about the risk of malformations with a particular treatment. But again, that effect might be mediated by increase in multiples and an increase in risk due to that. And we could also ask sort of like, what is the effect of treatments if we could intervene to eliminate multiples? Again, though, this requires sort of quite strong assumptions if we're thinking that this is some sort of causal estimate. But this sort of restriction to infants approach does lead to a slightly different sort of estimand than the other approaches we had.